maybe. Well, today we'll be talking about evolution of populations, which is the level at which evolution actually occurs. So, because individuals do not evolve, individuals can be, either a little more adapted to the environment or a little less adapted to the environment, but none of it matters until they're reproducing. So, as long as this little thing starts working. All right. Okay, so evolution of populations. There's Darwin's finch, which is just super famous. All right, so these are the learning objectives. Hopefully by the time we're done, you'll have a decent understanding of these objectives. But first off, we're going to hit up a little review of genetics in order to more effectively talk about how evolution occurs in populations. So, relatively simple stuff. A gene is a discrete unit of hereditary information. So, basically, a gene codes for a protein, and so you have various different genes, thousands of different genes in a genome. Each gene affects the animal in some way, and genes are passed on to the next generation. So genes are really the unit of inheritance, and we'll kick up a little review of good old Mendel, Gregor Mendel, and Mendelian genetics. So he had a little bit of vocab. We have the allele. The allele is alternate versions of genes. So you have gene 1, say it's for, hair color, and you'll have a gene that represents blonde, and a gene that represents brown, and a gene that represents red, and those are all alternate versions of gene 1, so they're alleles. So, none of that works. By the way, those aren't actual alleles that exist in humanity. As far as Mendelian genetics, in all my research, there was only one that actually turned out to be true Mendelian genetics, and that's the type of earwax you got. So if you've got wet, orangey, yellowy earwax, that's a dominant trait, which we represent with capital letters. So capital letters are a dominant allele. And then if you've got crumbly, dry, whitish gray earwax, that's a recessive allele. So dominant alleles are always expressed, whereas if you're going to express a recessive allele, both copies have to be recessive. So we talk about an organism's genotype. That is the sum of the alleles present in the organism. So if we're looking at earwax, right? Phenotype is what you see, the physical manifestation, physical appearance. That's phenotype. So that would be wet earwax or dry earwax. And the genotype is just the allele combination. If you have wet earwax, you could be dominant in two separate ways, right? And so we use homozygous dominant, which is which one of those? Yeah, top one, the pair of capital letters for it. And then we have heterozygous. Both of these express the dominant phenotype, but they are separate genotypes. And then, of course, there's homozygous recessive, which is the only way you can express the recessive phenotype. So, makes sense to everyone? I can always kick into it a little more. Okay, good. So, little genetics reviews. So, I talked about phenotype already. It's the observable traits, physical appearance, phenotype. And so dominant phenotype is either homozygous dominant or heterozygous. Recessive phenotype is always homozygous recessive. And this is for Mendelian genetics. A lot of people like to throw off eye color because it's flashy, you know, brown versus blue, but that's governed by multiple genes. So it's not basic Mendelian genetics. So as I said, earwax, the only basic Mendelian genetics ones. You tend to see wet earwax in any populations derived from Europeans, and dry earwax tends to be more common in Asian populations. So, all right. So the grist for natural selection is genetic variation. So genetic variation represents the differences among individuals in their genes. So when we talk about genetic variation, we're primarily, we're referring to the heritable part of the organism. So genetic variation is what's inherited. For natural selection to have an effect, you have to have variance in the genes within the population, so that some genes are favored over other genes, that some genes become more common in the population. That's evolution by natural selection. So genetic variation is the grist for that. That's what natural selection acts on, the varied genes in the population. We can actually contrast genetic variation with a concept called phenotypic plasticity. So these are variations in an observed trait, right? Phenotype, what you see. These are variations in an observed trait that are due to environmental influences, not genetic. So in this case, we have two separate caterpillars. They are the same species, Nemoria erickssonaria, and they eat separate food sources. A and B have been eating on separate food sources. A has been eating oak flowers. B has been eating oak leaves. By virtue of their diet, they have produced separate phenotypes, not by virtue of some genetic change. So these actually have the same phenotype, the same genotype, but they look different because of their environment. So oak flowers produce a phenotype that's better camouflaged to the oak flowers, whereas eating oak leaves produce a phenotype better camouflaged to the stems or the branches. So that is not inherited. So if you get two of these together, the oak leaf eating caterpillars, and you mate them, and then you put their offspring on oak leaves or oak flowers, they'll just develop the corresponding phenotype. So that's phenotypic plasticity. So the word plasticity just sort of refers to the ability to, bend or change or alter. So, So yeah, very cool. Phenotypic plasticity is not inherited, so the observed traits due to phenotypic and plasticity are not inherited. It's only true genetic variation that allows inheritance. So when we look at genetic variation, one of the things we like to calculate is average heterozygosity. Because heterozygotes are a pretty good reservoir of genetic variation, right? When it comes to simple Mendelian genetics, a heterozygote carries a dominant allele and a recessive allele. So mating heterozygotes can produce dominant individuals and recessive individuals. So when looking at the variation in a population, sometimes a good shorthand for that is heterozygosity. So It's used to quantify genetic variation in Mendelian traits. And it's literally the average percentage of loci that are heterozygous. Loci, if you don't remember, L-O-C-I, it's the plural of locus, and that's just the location where the trait is found in the genotype, or in the genes. So, if you have your DNA strand, there's the locus for gene 1, there's the locus for gene 2. So, it's the average percentage of heterozygous genes. So the higher percentage of heterozygotes in the population, the more genetic variation in the population. And again, this applies to Mendelian inheritance. Not super useful for quantifying genetic variation in human populations because we can do it based on earwax. But for other animals, there's a large number of separate genes that have Mendelian inheritance. So there are probably a large number of genes in humans that have Mendelian inheritance that govern internal conditions. And that's not easy to see. So, all right, so if we have 1,920 and 13,700 loci heterozygous, then we have 14% heterozygosity. So you can only know this through gene sequencing, or if you have something going on like codominance, where the heterozygotes have a different appearance. So, all right, when we look at populations, we can compare genetic variation between separate occurrences of these populations of organisms, all right? So, for instance, we have house mice, Mus musculus, and we have this island where we have a pretty decent mountain range blocking these populations from intermingling. And so we can measure the genetic variation within each population and compare the variation between the two. So because these mouse populations are isolated, we've seen patterns of fusion that are different. So these populations are breeding only within their own population. Blue breeds with blue, red breeds with red. And so we've seen fusion of chromosomes in different places in the population. So, yeah, you got a fair amount of genetic variation between the populations, which is pretty darn cool. So, genetic variations between the populations is when we measure geographic variation. So we're measuring populations separated by geography. And so when we look at those genetic variations, oftentimes we'll talk about a cline. A cline is a graded change in character based on geographic location. So our example with these fish here. This is a pretty good example of a cline. We're looking at a graded, a shifting change in variation between populations based on geography. So, this example here is pretty cool. What we've got here is the mummichog fish. I don't quite know how to say that correctly. When it comes to species names, I just say things with confidence and hope it works out from there. So, mummichog fish. So, lactase dehydrogenase B, which is an enzyme that functions in metabolism, helps fish catalyze metabolic production in cold temperatures. So if we look at the frequency of that allele in populations based on geography, in Maine, where the average temperature of the waters is relatively cold, we see a high frequency of that enzyme. And then it goes down as the water gets warmer because it's unnecessary. So that's a cline. where we see a decrease over geographic area. So again, we have our little house mice and our mummy-chawed fish. So the house mice are not an example of a climb. They're an example of just straight geographic variation in populations because we're looking at fusion events. So we have population 1 and 2. Can't really put that on a graph and have a graded change. We're looking at fusion events in the separate populations. So that's just straight geographic variation, whereas our mummy-chawed fish is a great example of a climb. So we can see all this variation between separate populations. Then the question becomes, how does variation arise in populations? Well, there are 4 basic sources of genetic variation. First off, one you probably all know. Who wants to take a guess?
Mutation.
There you go, mutation. Right? So genetic mutation is one source of variation within a population. It's a change in nucleotide sequence. So, change in nucleotide sequence. That is mutation. It does not occur super often when looking at larger animals. Remember when I was talking about bacteria, they have a relatively error-prone DNA replication system, and they reproduce quite often. So mutation occurs a lot more in bacteria. When you get it to the level of animals such as mammals, you really only see mutation apparent in a population in reproductive events. That's when it passes on into the population. So it does not occur as often, and animals have a lot of error checking in their replication mechanisms. So mutation isn't as big a source of genetic variation for animals as it is for bacteria, which is pretty cool. The largest proportion of all mutations are either neutral, meaning they have no effect on survivability or reproductive success, or they're negative and tend to decrease in the population because they have a negative effect on survivability or reproductive success. Rarely you get beneficial mutations. This is because mutations don't tend to cause huge changes in an organism. You're not going to get a mutation causing, eye lasers. X-Men. Anyway. These can cause, primarily they'll cause small changes, mostly unnoticeable changes. But if you chart through history, once you start adding evolutionary time to it, and you're tracking thousands of years, you can get mutations that occur every once in a while that actually have a dramatic effect. I'm not remembering the rodent, but in the Badlands of South Dakota, there's a rodent species where a mutation produced an entirely different alternate fur color. And that allowed the mouse to exploit a separate geographic area. So it provided camouflage in an area where before their coat color did not provide camouflage. So that mutation happened to have a dramatic effect. Most mutations do not. So all you got to add is thousands of years, which, as I mentioned earlier, our brains suck at comprehending. There are other sources of genetic variation. You might write all of these down off of the one I posted online, but deletion, duplication, translocation, insertion, inversion, and isochromosome. These are alterations in the chromosome itself, not in a small area of nucleotides within a single gene, but rather a big alteration of chromosomes. So, deletion is when you lose one or more genes due to an event during replication. Duplication is where you get an additional copy of a gene. Translocation is where a gene changes locations. I've got a picture on the next slide that is on, your lecture online. So hopefully that's pretty self-explanatory. But translocation is where it changes position. So it moves from one chromatid to another. It may be another, right within the pair, or it may be another on a completely separate chromosome pair. And then we have inversion, or insertion and inversion. I don't want to explain all of these right now. Anyway, insertion is, I might as well. So, here, you know what? I'll just pop over to this slide since it's got the nice pictures. So, insertion is where a portion of one chromosome is deleted from its normal place and inserted into another. So, deletion is where we completely lose the genetic material, right? Insertion is where we lose the material from one chromosome, but add it to another. So it's inserted onto another chromosome. Translocation is where it changes position from one chromosome to another. Inversion is where it reverses position. Isochromosomes are kind of funky. Isochromosomes are where we separate out the chromosome and we get, if you have your chromosome, it splits at the chromatid there, and it becomes two separate chromosomes that are each a chromatid from the original, which is pretty funky. I'm not going to emphasize this set too much on an exam, but it's worth knowing that there are a variety of ways to get big changes. So this is how we get larger changes when we look at an organism. So going back, rapid reproduction is a big one. As I was saying with bacteria, the more often you reproduce, the more often you get mutations that can proliferate into the population. So, more reproduction you have, the bigger the impact of mutation. And then a great source of genetic variation is, in fact, sex. So, you were in Bio 1, you talked about sex, which is where you produce a haploid set of chromosomes, a sex cell, then another one produces a haploid set of chromosomes with their sex cell, and you combine it. So you get a random assortment of genes from one organism and a random assortment of genes from another individual. They combine, and you get a new combination of genes. So that right there produces a fair amount of genetic variability with individuals in the population, just straight sex. But in addition to that, fertilization, straight sex, produces genetic variation. In addition to that, you may have talked about crossing over in Bio 1, where the chromatids tangle up, and that causes a snap, and then they switch positions. So that right there causes genetic variation. There's something like 70, on average, 70 instances of crossing over in women when producing a sex cell, and something like 30 instances of crossing over in men when they produce a sex cell. So it's very common. And then independent assortment of alleles. When I said, you take a random shuffling of the alleles from one parent, put it in. So that's independent assortment, because if you have a pair, right, so this is a pair for chromosome 1. One of these is, the chromosome inherited from mom, and one of the chromosome inherited from dad, and there's basically a 50-50 chance of this chromosome ending up in the sex cell. That's independent assortment. So all sorts of stuff producing random variation, increasing genetic variation in the population. New combinations combined with crossing over events may produce new variation that may impact survivability and reproductive success, which is then acted upon by natural selection. So, genetic variation is the grist on which natural selection acts. And now we'll talk about some examples of genetic variation affecting populations, specifically mutation. So we're looking at stickleback fish. And stickleback fish, you have marine sticklebacks and freshwater sticklebacks. Freshwater sticklebacks are generally, based on evidence we have, evolved from marine populations. And different freshwater stickleback populations, on different continents, evolved from different marine populations. So it was multiple evolution of this to get different freshwater sticklebacks on different continents. Mutation being a source of genetic variation. So I said most are harmful or neutral. Wow, I've been through this. So some are beneficial and can even have dramatic effects. I talked about the rodent that I can't remember which one it was, probably some species of mouse in the Badlands. And then this is the marine stickleback. So marine one has a large pelvic hind fin and heavy armor along it. And it seems like every time they hit freshwater, they lose the pelvic fin and they lose a lot of that armor. So the hind fin skeleton alterations are controlled by alterations in the activity of PIDX1. So just mutation in the PIDX1 gene can produce a dramatic effect on the pelvic fins. So another example of mutation producing a relatively dramatic effect. Okay, so apparently I forgot to add my little transitions here. So before scribbling down absolutely everything on here, let's go through it one at a time together. So persistence of a gene explains why specific frequencies sort of persist in a population. So when we talk about persistence, we're talking about well, I hate defining something with word, but specific combinations of genes, specific genotypes or phenotypes staying relatively constant in a population. So when we say wild type, so there's this phrase called wild type, and we apply it to animals. The wild type is the type that you see in the environment. So remember when we talked about wolves, they all have the same general appearance, rounded ears, sort of a longer snout, larger size than domestic dogs. But when we bred them for artificial selection, we uncovered a large reservoir of genetic variation. So the persistence of the phenotype, that wild type phenotype, was based on the adaptation to their environment. So adaptation tends to produce persistence, where you get a specific set of alleles, a specific phenotype that is well adapted to the local environment, and so they persist in the local environment. Hardy-Weinberg is a great equation. So the Hardy-Weinberg principle governs a set of hypotheses that we can examine in order to determine whether or not evolution is occurring with regards to a specific allele, right? So, or a phenotype. So, say you've got coat color, right? You want to find out if one coat color is favored over another. Well, we can compare frequency of those coat colors in the population to our Hardy-Weinberg principles. So, in order for an allele to not be evolving in a population, we have to satisfy a specific set of conditions. One, we have to have a large population. A large population is more resistant to natural selection. So the larger the population, the smaller, generally, the change induced by natural selection. The more resistant it is to sudden events. For instance, a disaster. A very large population, if there's a disaster that wipes out a certain number of them, that larger population will be a reservoir for maintaining the original genetic variation. So at some point in the past, cheetahs went through some event, we're not sure of what it was, and they lost something like 90 to 95% of all their original population. And so the genetic variation present in all cheetahs is just what's left. So a large population is able to resist big changes like that, random events that wipe out a large number. Random mating. In other words, mates are not chosen based on that phenotype. So a mate is not chosen based on that particular phenotype. No mutations. So the trait is not continuing to be altered by subsequent mutation events. So mutations could occur and not violate Hardy-Weinberg if they're neutral. So if they're neutral mutations not affecting the phenotype, then you may not have, it won't violate Hardy-Weinberg. But in general, when we say no mutations that cause natural selection to act on the allele. No gene flow. So when we talk about gene flow, basically we're referring to the transmission of genes between populations. So if you've got population 1 in area 1 and population 2 in area 2, if they can meet up with each other, they can transfer genes between the population. So when we say no gene flow, we say that gene flow is not affecting the frequency of that allele or phenotype. So gene flow is immigration with an I, where new individuals enter the population. And then we've got emigration, can't remember if there's two M's. It's not English. Where you have individuals exiting the population. E for exit. Which isn't all that helpful when there's an E for immigration. I've never been a fan of slight pronunciation differences, meaning the exact opposite thing. Immigration, emigration. So I'll specify whether it's an I or an E if I mention it again. But basically, there's either no gene flow, so new populations aren't introducing new variations of that allele. So no gene flow is when you don't have any individuals moving between populations, or you may have gene flow that's not affecting the allele frequency. So they have the same, the two populations have the same frequency, so randomly individuals may cross between, but it doesn't affect the allele frequency. A lot of stuff here. And then finally, there's no selection occurring with that phenotype. So that phenotype must have no effect on survivability and reproductive success. So these are the conditions under which evolution does not occur in a phenotype. So, large population resistant to sudden changes from like a disaster or something. Random mating, mates are not chosen based on that phenotype. Mutations are not producing a source of variation. Gene flow is not altering the frequency, and natural selection is not altering the frequency of those alleles. If you satisfy all of these conditions, then we can say the gene is at Hardy-Weinberg equilibrium. So, Hardy-Weinberg equilibrium. So we actually have a mathematical formula from which we can determine whether or not evolution is occurring within a set of alleles. That gives us sort of a baseline of comparison. We can plug our data into the equation and then compare our data to the data found in the actual wild population and see if there's a difference between the two. So if there's a significant difference between what you calculate in Hardy-Weinberg and what's present in the population, you could say that evolution is occurring. So it's almost a basis for statistical comparison to determine whether or not evolution is occurring in that particular phenotype. So, one of the mistakes people made, like I guess in undergrad I made the mistake, is to assume when we say Hardy-Weinberg, we're saying the entire animal population isn't evolving. And we're specifically generally referencing like a phenotype. So in squirrels, maybe brown coats and black coats. So, you know, we're not measuring whether or not evolution is occurring in the entire organism. So, good old-fashioned Hardy-Weinberg formula. So Hardy-Weinberg is based on Mendelian genetics. And our base Hardy-Weinberg formula is P plus Q equals 1. P is all dominant alleles in the population. And Q, who wants to take a shot at what Q is? All the recessive alleles in the population. All right, so I'm underlining all. If we have a population of individuals, right, and we calculate out the number of dominant phenotypes we see and the number of recessive phenotypes we see. So we look at, I don't know, a mouse, and we see in this particular population 125 brown mice, and we determine that the brown coat color is dominant, and let's say 75 white mice, which is recessive. Can we plug 75 white mice into Q? Can we plug the frequency of white mice into Q?
You don't know if the brown mice have recessive.
Yeah, there it is. So these 125 brown mice can be homozygous dominant, or they can be heterozygous dominant. And if they're heterozygous, they have a recessive allele. So Q is all the recessives from the homozygous recessive and the heterozygous. And just like P, all the dominant alleles are from the homozygous dominant and the heterozygous. So P plus Q equals 1 is not incredibly useful when just looking at raw data from the population. Luckily, we have an extension of Hardy-Weinberg. in which we have p squared plus 2pq plus q squared equals 1. So now we have 3 variations. So what do you think p squared represents?
Dominant.
Yeah, p squared represents the homozygous dominant. How about 2pq? There we go, the heterozygous, because P is dominant alleles, Q is recessive alleles, so we've got a P and a Q. That's heterozygous. 2pq is our heterozygotes. And Q squared by process of elimination, there you go. The homozygous recessive. Between these two, we can then use population data to get a full Hardy-Weinberg equilibrium calculation. So, first off, our variables here, we're going to be plugging data into them that's frequency. So when P plus Q equals 1, that 1 represents 100% of the alleles in the population. So when we have our 75 white mice showing the recessive trait, we know that what genotype are they? Homozygous recessive. Only way to show the recessive trait. So they are homozygous recessive. There's a total of 200 individuals in the population. So we can plug the frequency of homozygous recessive into which variable? Q squared. Good, good. And what we're actually plugging into there is 75 divided by 200, which should be something like 0.375, if my math works, because dividing them out is the actual decimal frequency. So if you were to convert that into percentage, 37.5% are homozygous recessive. So now we have Q squared equals 0.375. Can we get all the rest of Hardy-Weinberg filled in from Q squared? Didn't know you'd have to do algebra, did you? There you go. We can root Q squared to get Q. So, root 0.375 equals Q. I'm not doing that in my head. Anybody got a calculator handy? Or, you know, some sort of math wunderkind? I don't know. 0.612. Okay, so Q equals 0.612. All right. So now, 0.612. Since P plus Q equals 1, we have 0.388 for P. Boom. And now we can use P and Q to fill in all of our P squared plus 2PQ plus Q squared equals 1. So, who wants to square P for me? Anybody? Square P. Square of 0.338 or 0.388. 0.151? Okay. 0.151. 2 times P times Q. Anybody? .474. Okay. So there we go. These are the frequencies we would expect under Hardy-Weinberg equilibrium. So if Hardy-Weinberg equilibrium is occurring in this population, then 15.1% of the population would be homozygous dominant, or 30 individuals. Then we would expect 47.4% to be heterozygous, which is, what is that, 95 individuals. I don't know if this is going to perfectly come out. And then 37.5% to be homozygous recessive, which is our 75. Hey, that worked out. So that brings us to our 200 individuals. So in order to determine whether or not this population is at Hardy-Weinberg equilibrium, we would then need to be able to sequence the population's genotypes. So if we sequence that population's genotypes and their frequencies are close to these, then that would be some compelling evidence that they're at Hardy-Weinberg equilibrium. But if you've got something else going on, say there's 60 homozygous dominant and, you know, what is that? 35, or no, 65 heterozygous. That's a pretty big change from what we would expect under equilibrium. So that might be evidence that natural selection is acting on this allele combination in some way. So natural selection is altering allele frequencies. Maybe it's through mate choice, maybe it's through pure natural selection, but something is occurring that is altering frequencies away from what you would expect at Hardy-Weinberg equilibrium. So it's a baseline for whether or not evolution is occurring with respect to a specific allele. Does that make sense? Okay, sweet. And I got ahead of myself, so alleles. So we've got the Hardy-Weinberg equilibrium formula for just total number of alleles. And then we've got the Hardy-Weinberg equilibrium formula for genotypes, p squared plus 2pq squared, 2p cubed plus q squared, where we can then separate it out into homozygous dominant, heterozygous, and homozygous recessive. Hopefully you've written all of this down because I wrote it all on the board. Yeah? Sweet? Okay. So then I went through an example, which we've also already done. So we had earwax type, wet versus dry. So this is straight Mendelian dominance in humans. And we had 320 and 180, producing a population of 500. So is evolution occurring with regards to earwax? We do Hardy-Weinberg equilibrium in order to determine our baseline that we can compare to the population. So again, we convert that raw number into frequency data. 0.36 is the frequency of individuals that are homozygous recessive. So we plug that into which one? Q squared. That's right. Q squared. So why isn't it Q? That's right. It's not Q because of heterozygous individuals. Because P plus Q equals 1 is the total raw number of all alleles, all dominant and all recessive. Heterozygous are dominant individuals that carry a dominant allele and a recessive allele. So Q is all the homozygous recessive alleles plus all the recessive alleles from heterozygotes. So 0.36 cannot be Q, so it is Q squared. We root it in order to get Q. From there, we can get P, and from that, we can fill in the entire rest of the equation. So there's our genotype equation for Hardy-Weinberg. Boom. So 16%. If we got 500 individuals, we should see 80 homozygous dominant, 240 heterozygous, and 180 homozygous recessive, which we do see because that's what we based our entire formula on. And then if we look at the actual, here we've got something way off. So what can we say when we sequence it and see this sort of frequency compared to our Hardy-Weinberg calculations? It's not at equilibrium, which means what is occurring? Evolution. There you go. So that is Hardy-Weinberg equilibrium. So that's algebra, and it sucks that you have to take algebra in biology, but it's very useful as a comparison tool. And today's lab is on Hardy-Weinberg equilibrium. So this is another calculation thing, but same thing. You do the frequency, plug it in for Q squared, root it for Q, get your P, fill in the rest of it. Okay, so new mutations are a mechanism for evolutionary change. So new mutations can result in phenotypes that are acted upon by natural selection. Gene flow can be a mechanism for evolutionary change. So if individuals from another population bring in new genes that affect survivability and reproductive success, then they can be acted upon by natural selection. Non-random mating, which means in order to mate, one of them is looking for a specific trait, a specific phenotype. So mating, your chance of getting a mate is governed by phenotype. Right? So think peacock. Those peacocks have that big, beautiful tail, and the females look for the biggest, most beautiful tail in order to mate. So that phenotype is non-random mating. So, you got to have the biggest, most beautiful tail in order to have the highest reproductive success. Genetic drift is when you have random events altering gene frequency in a population. And then good old-fashioned natural selection, which is primarily based on environment. So, gene flow is the movement of alleles from one population to another, which I've said before. There are actually three basic modes for gene flow. I already mentioned immigration and emigration. So, immigration is new genes entering the population. Emigration is genes exiting the population. So, if you've got population 1 and 2, here's population 1, and here's population 2. Genes can immigrate into population 1, and genes from population 1 can emigrate into population 2. That's useful for organisms that can move on their own. So, pollination, number 2, so 1 is immigration, emigration. Number 2 is pollination or other vectors. So, in plants, when we look at pollination, for a lot of them, it's governed by wind. And then there's all the flowering plants that that tend to have an insect vector. Insects pick up pollen and transfer it to other individuals. So you get gene flow between populations based on non-physical conditions. For organisms in the ocean that cannot really fight the current on their own, the current is a vector. So number 2 is pollination. Vectors like wind. And 3 is basic movement of gametes. So especially important in marine organisms. So if you've got your little sea anemone here, fantastic drawing. Sea anemones, they find a spot to hang out and they live their adult life there, right? And so, in order for gene flow to occur, they release their little gametes, and their gametes move around. And then current can take them around. Current can also take larvae around. So you've got gene flow acting by 3 separate mechanisms. 1 is immigration-emigration for organisms that can move on their own. 2 is vectors, like pollination from wind or insects. 3 is movement of gametes or larvae within current, basically. So, those are 3 mechanisms for gene flow. We like to use animals a lot as examples. So this is Parus major, the great tit. Giggle all you want. So the great tit, we can examine their populations on Vlieland, an isle in the Netherlands. So our eastern population tends to be well adapted to island life. But our central population gets a lot of immigration from the coast. So what happens is those coastal birds roll in, immigrate into the central population, and bring genes that are well adapted to coastal life. Those genes may be maladaptive to living on an island. So what we see when we measure survivability and reproductive success, so this one is just straight survivability, females in the eastern population, born into the eastern population, have much higher reproductive success than females born into the central population. That's because females born into the central population have a much higher chance of getting genes from coastal birds that are not as well adapted. So having a higher chance of getting maladaptive genes that have a negative effect on survivability and reproductive success, is borne out in this graph. So immigration, gene flow, affecting survivability and reproductive success. So, boom. Great example with the great tits. Then we have non-random mating. There are sort of 2 basic mechanisms for non-random mating. We have assortative mating and disassortative mating. Leaf beetles tend to practice assortative mating. What that means is that a female leaf beetle will look for a male that has a similar phenotype. So, if you have 2 males, one with a similar phenotype and one with a dissimilar phenotype, she will choose the male with the similar phenotype. This tends to increase the number of homozygotes in the population. Why would it do that? Well, yeah, there's reproductive success is what you're looking for. They have higher reproductive success. So, homozygous dominant produce offspring that are homozygous dominant. So when a homozygous dominant female mates with a homozygous dominant male, they're going to have offspring that have that homozygous phenotype that other females are looking for. And then homozygous recessive females are going to be looking for homozygous recessive males. So because heterozygotes can produce a mix of offspring, you might not have as high a reproductive success, especially if there's codominance or incomplete dominance, where heterozygotes have either a mixture of those phenotypes or both phenotypes expressed at the same time. So assortative mating is where mating, similar individuals are favored, and it increases the number of homozygotes. So if assortative mating increases the number of homozygotes, disassortative mating increases the number of heterozygotes. And if assortative mating is where they're looking to mate with a phenotypically similar individual, disassortative is where they're looking to mate with a phenotypically different individual. Yep. So pigeons choose mates that have plumage different from themselves. That's why when you roll out to a Auraria station, no two pigeons look the same. Female pigeons are going to choose a male that has different plumage. So, the more variation in plumage you have, the more likely you are to find a mate. And heterozygotes tend to be a reservoir for increased genetic variation. So, disassortative mating, choosing phenotypically dissimilar individuals, phenotypically different individuals, increases the number of heterozygotes. All right, genetic drift. So genetic drift is changes in the gene pool due to chance events. So rather than individuals favoring some trait or the environment favoring some trait, a random event occurs that alters genetic variation. So, this is most important in small populations. If you have a small population and an earthquake occurs and half of them die, you're going to have a much larger impact than if you have a very large population. Because that very large population will be all over the place. And that earthquake may only kill off a small number of them. So assuming that phenotypic variation is relatively stable in the rest of the population, that random chance isn't going to have as large an effect. So genetic drift has a greater effect on small populations. There are often two examples we'll use when we talk about genetic drift: the bottleneck effect and the founder effect. So, the bottleneck effect. This is when a big disaster, such as a flood, occurs. Maybe an earthquake, a fire, humans rolling in and being jerks, killing a bunch of stuff. So the bottleneck effect is where you have a relatively genetically diverse parent population, and something happens which kills off the grand majority. We call it the bottleneck because it's a decent metaphor. If you fill a bottle with beads, different colors representing different phenotypes, and then you shake that bottle into a little beaker, only a few come out, right? Because that bottleneck is there, preventing a bunch of them from coming out. So what you have is the parent population, and then some event occurs, maybe an earthquake, whatever, fire, and only a few survive. So the surviving individuals are the basis for all of the genetic variation in the surviving population. this can produce dramatic reductions in genetic variation. Since there was a absolutely massive bottleneck effect on African cheetahs, most cheetahs, or all cheetahs now, are almost hilariously inbred. They have low cub survivability, which you would expect from a highly inbred population. Because if, this individual, and this individual, and this individual have recessive alleles that may impact survivability of offspring, when they breed, more individuals will have those recessive alleles, and as they breed, you're going to have it more likely for offspring produced from those matings to have those recessive alleles, which can produce increased loss of cubs. A lot of stillborns, low survivability. In addition to that, if you want a really great example of just how little genetic variation is present in cheetahs, it's thought that if you were to take a skin graft from any random cheetah, you could give it to any other random cheetah and it wouldn't reject. I don't know if anybody's ever done that experiment. Just like, hey, let's skin cheetahs. But that is the bottleneck effect. So, talked about cheetahs. Then we have elephant seals. Good old white people thought elephant seals were great for eatings and hunting and various commercial uses. And by the 1890s, we had hunted northern elephant seals to about 20 individuals. And then someone said, hey, maybe not drive them extinct. And so now there's about more than 30,000 northern elephant seals, all descended from those 20 individuals. So all the genetic variation present in the modern population is the genetic variation that was present in those 20. So who knows what genetic variation was lost in that bottleneck. Our southern elephant seal here wasn't hunted to near extinction. And so they have much greater reservoirs of genetic variation. And then I talked about the cheetahs. So their bottleneck effect was like 10,000 years ago. And then there was another bottleneck effect thanks to white folks when we hunted them to near extinction in the 1900s. So they are incredibly reduced variation. We can contrast that with the founder effect. So, whereas a bottleneck effect is some event that kills off a large number of individuals, resulting in reduced genetic variation in the population, a founder effect is where a small number of individuals create a new population. A small number of individuals found a new population. So you have that original population with large genetic variation, and then a small number of them find a new place and found that new population. And so all of the genetic variation descending from those founders is what's present in the population. So we have Tristan de, I'm going to say Cunha, I don't know. So in 1814, Tristan de Cunha was founded by 15 people. 15. And so it's the small island. It's relatively out of the way. So not a ton of Europeans or anybody was saying, hey, let's go join the colony. And so most of the population present at this point, because they were 1,500 miles away from the nearest continent, the population of this island is pretty much descended from those 15 people. One of those 15 colonists had a recessive allele for a type of blindness. Today, 1 in 60 people have the progressive blindness, and 9 of them are known to be carriers, which means they're heterozygotes, carrying one of the alleles. So it's relatively common, incredibly common when you compare it to the original population, right? So Great Britain is where they originally came from, those 15 folks. So the modern population, what's left from 1814, 1 in 60 have progressive blindness. In the UK, 1 in 4,000 have progressive blindness. So because we have such a small founding population, we have reduced genetic variation. So, genetic drift. In bottleneck effect, some random event occurs that drastically reduces population, so the survivors are the remaining genetic variation. It reduces genetic variation through killing off a large number. The founder effect, we have low genetic variation because we had a low number of population founders. So, and then we got 4 minutes left. I'm going to run all the way up to 9:15. We have natural selection. So we were talking about this all last lecture. So I have this picture of crows favoring green beetles. So if the crows are favoring green beetles, how do you think it's going to impact the phenotype of beetles in the population?
They're more. The ones that are orange are going to survive, so there'll be more of them.
Right. Because the crows are preferentially feeding on green beetles, you're going to see increased frequencies of orange beetles in that population because they're less resistant to predation, which means there's more of them around, which means they're more likely to be able to reproduce successfully. So orange, the orange phenotype will increase in the population. So natural selection is based on what traits lead to reproductive success, or traits that negatively impact reproductive success and survivability. I like to primarily talk about reproductive success. As I said, Monday, survivability don't mean much if you're not mating. So, traits that have an impact on reproductive success. So basically, in natural selection, something in the environment, whether it's predators, whether it's competition for a food source, whether it's the color of the surrounding vegetation, the amount of water available, something in the environment is having an effect on the population. We call those things selective pressures. So something is exerting a selective pressure on a population. Some individuals may carry a trait that allows them to have more increased survivability and reproductive success. Right? They spend less energy trying to survive in the population. They spend less energy trying to survive and reproduce when exposed to that pressure. So because they spend less energy there, they have more left over for reproduction, so they reproduce more effectively than individuals who don't have the trait. So the trait that is adaptive becomes more common. That's natural selection producing evolutionary change. So we have some terminology here. We'll see how much I can get in a minute. So, directional selection. If we have this frequency of individuals originally, right, we can look at this picture here, the pigment in these moths. Which moths do you think are more likely to survive and reproduce? The brown ones. Why? So they're able to resist what? Predation. Right. So directional selection is when we select for one trait over the other. So we go from this bell curve of frequency towards 1. So it favors one extreme. So that tends to cause a huge effect. It tends to eliminate the other extreme. You can imagine how that works with those moths. Those white moths are easy pickings for predators. And so they may very quickly be eliminated from the population. So directional selection is where we favor one extreme over the other. And it's 9:16. So, sorry for keeping you late. And those of you who have my lab, I'll see you in like 3.5 hours. And if you have any question over Monday or today's lecture, I have office hours at Science 2057 from now until 1 o'clock, essentially.